Okay, so I'm still waiting for a couple of you that have blank whiteboards. So click off and then on. Apparently, if you don't click it, I can't see it. So even if you're typing it, until you click it, I don't see anything on your board. So I see April. I don't see anything on Briquel or Brianna yet. I see Eli. I don't see anything on Emma. Evan, Philip has stuff. Joseph, nothing. Maddie and Megan, I see something. Good. Natalie, yes. Neil, yes. Another thing that I'll do a lot of shorthand, and you'll see this in PowerPoints, you talk about tissues all year long, is you'll see abbreviated on a lot of the PowerPoints in different spots, a capital C and a capital T, and that's connective tissue. Okay, so you're going to see things referred to with just their two capital letters. So you guys are welcome to use some of those abbreviations as well. Once we have these tissues down, I'm going to save your whiteboards real quick. Tissue 10132020. We have to have evidence that we really do try to teach you things. And I just took proof of it. All right. Next. So most of you are getting that down. If on your whiteboard, you could put if you're at a 4, 3, 2, or 1 level, 4 being you could teach it in some original creative way. Beck, I think you just have to click on it. The last part of the code that's on the stream is on the actual stream page of the attachment. So it's Y6X6D. So if you're at a 4, if you can teach it original and creative way, 3, if you're proficient at it, like you can do it on whiteboards or answer Kahoot questions. 2, developing, there's one of them you keep forgetting. And 1, you were not aware that this was a target for chapter 5. So I'm seeing some 3s and 4s, a couple 2s, okay. 3, okay. So moving on. A reminder that there is a study slide. So this slide has everything on one slide. If you could fill in a chart like this for this target, you would be exemplary. Then there's another one where you would be just dragging it around and mixing and matching it, which is kind of like proficient level. So you can practice both of those to get down all of the information on the tissue types and their functions, locations, and what distinguishes one from the other. The second thing that we talked about last week was this idea of being familiar with the cut of tissue, so the chunk of tissue and what direction they took the chunk. So when you go in for a biopsy, they are taking a little piece of you, which is a tissue because it's made up of multiple cells. And when we look at some of these tissues under a microscope, and your book has the lab for this is color pictures of tissue samples. But you have to keep in mind is what section was it? Was it a cross section or a longitudinal section? Because it'll look different. And that just takes practice and paying attention to the label. So the other thing that we did last week was to name characteristics of epithelial tissue. So if you're looking at an unknown slide, how will you know it's epithelial tissue? What are some characteristics that that tissue has that muscle, nervous, and connective tissue don't? So give me at least 3 of those. Your book goes through about 8 or 9, it looks like. And add that to your slide. What are some characteristics of epithelial tissue? I see a fish and a smiley face and a sailboat. Okay, good. Now I see Maddie has some real ones. Good. Good, Lauren. Grace, good. Some of these are really important for functions that we're going to talk about, right? So I see some of you putting no blood, avascular, which means they have to get all their nutrients through diffusion. And if you get too far away, those cells are dead, like in the skin. Once we get to the top of the skin, the cells don't have the ability to even get nutrition. And so they slowly fill with a protein called keratin and die. Good. I see someone else with tightly packed cells. Yes, so highly cellular. When we look at connective tissue, like especially bone tissue, there's hardly any cells. They're just kind of dotting it throughout. But this is like layers and layers of tightly packed cells in epithelium. Okay, I see acts as a barrier, secretes things, yes, and absorbs. Protects, covers body surfaces, good. So talking about locations. Widespread, yes, this is throughout your body. It's all over, it's what your skin is epithelial tissue, and then anything that's lining a blood vessel, anything that's lining an organ, all of those are also epithelial tissues. Raquel, I'm not seeing anything on your whiteboard yet. Capture, I'm going to capture this just to talk that I do talk about worst reason twos. All right, let's look at these. So again, these are things you should already have notes on, and so you should be doing whiteboards on them. If you don't know some of these, make sure you go back and say 5.2 or something on your notes. It just tells you where to go back in your textbook to find more information. So I saw a lot of you putting widespread on the body, throughout the body, covering these body surfaces and lining cavities. One of the ways, though, under the microscope, if you're given a unknown slide, which is what we normally do in the classroom, is look for that free surface. So you're going to look for where there's just clearness, where there's no cells and no tissue, no staining, because that's unique to epithelial tissue, because it's lining something. So there's going to be nothing and then something, which are cells. And so these things all anchor to other things, like other types of connective tissue. And you guys said tightly packed cells. The one thing I didn't see, I don't think I saw from anyone, but it's important, is they readily divide. So they make copies of themselves. So we have this basal layer, basal cells that are on the bottom, and they readily go through mitosis. So they're always making new cells for you. So there's a kind of an adult stem cell down there. And when that cell gets out of control is when we see skin cancer. So it'll be important later that we understand that that does undergo mitosis. All right, a couple vocab terms. When something is thin and flattened or scale-like, we call it this word. This is going to talk again about epithelial tissues in terms of what the cells themselves look like. And then we're going to talk about how in your skin, for the integumentary system, the cells are in layers. So those layers have different cells and different shapes that are characteristic for that cell as it moves up and then gets sloughed off your body. So you're constantly shedding dead epithelial cells everywhere. And as they move up, they die, they fill with keratin, and they flatten out and then come off. And that takes about 30 to 40 days. Good, I see Maddie. And Emma, good. April. Still waiting for a couple of you up there that have blank whiteboards. There's a few of you so these are the people I can see that are pressing enter. April, Eli, Emma, Evan, that know this. If you don't know it, that's a different story. Joseph, Maddie, Natalie, Neil, Rain, Tommy, Adam, Emily, Grace, Lauren, Nushi. Those are all people that have something on their whiteboard. Yes, and squamous or squamous is the right word. We're going to talk about squamous basal cell carcinomas versus squamous cell carcinomas. So there's kind of two different places there in the skin that form skin cancers. The bottom layer, which is the most common, gets out of control because that's its normal job, is always to divide. And then there's these flatter ones that are called the squamous or squamous cells. And then stratified. So we have these layers, or strata, same term that's used in geology. All right. And then your book goes through a whole bunch of different types of cells of the epithelium. When we are back in the classroom, you guys are going to make a squamous cell prepared slide yourself of the inside of your cheek. The inside of your cheek or your cheek area is called buccal. So a buccal swab is also something you'd learn if you were in forensic science class. but you're going to prove to me that you're made up of cells and that you know how to use the microscope. So you'll do a smear, you'll stain it, and then you'll show it to me and label the cell parts. So the nucleus, the cell membrane, and the cytoplasm. Hopefully you will not be labeling any streptococcus bacteria, but you do have all sorts of bacteria in your mouth, but they're almost impossible to see with a compound light microscope. All right, so you'll see it says optional. So slides 17 to 24, if you get any of these questions right on your test, they are bonus. Okay, so I am not going to go through all these different shapes of epithelial cells. The only ones I'm going to go through are the ones we're going to need for when we talk about our skin. And that is these two words. So these two words talk about the epithelium again, and they mean flat and layered. So what were those words again? Oh, you guys haven't erased your whiteboards. So I'm just going to go ahead and show it. Those words mean stratified and squamous. But I keep mentioning a particular protein that these cells fill with that provide you waterproofing. So one of our lab activities will investigate these properties of our skin and how if you put water on it, it will bead up. And it's because of this protein. So what is the name of that protein? It starts with a K, that as you make cells in your skin, they fill with this dye and flatten and then come off. But it provides you this protection. Good, April. Anybody else know the word? It's also what your hair is made up of. So you see it on a lot of shampoo products. It's what your nails are made up of. These are all things that come out of your integumentary system and your epithelium. And it's interesting because they all use the same protein. Good, Evan. Natalie, good. Rain, I got it. Tommy, Grace, Emily, Paige, good. And Lauren. Okay, yes, it is called keratin. So you'll often see keratin as a product that people say are put in different things. Our skill number 2 that we'll be doing is me giving you slides of pieces of tissue from your skin and you telling me if it's normal or if it's cancerous and which type of cancer, which is a little harder, it is. So melanomas, melanocytes are cells that make melanin, so pigmented cells, so that's where your moles are. And oma means tumor or swelling. So melanomas are where a mole, right, starts to grow in a way that it shouldn't. That melanocyte gets out of control. It is the most deadly. It is not the most common, but it is most deadly and resistant to chemotherapy. So it's one of those you want to catch really quickly and early. And we'll talk about some ways to distinguish if you should go to a dermatologist about a mole. Basal cell carcinoma, the one in the middle here, is your most common, and that is named after the fact that it's a basal cell that got out of control. And then this is your squamous cell. I don't have this one as a prepared slide, so you'll be having to distinguish between these two on the left versus the one right below them, which is normal skin. All right, and this is showing you normal skin again. But we'll talk about that a lot when we get to our integumentary chapter. So slides 30 to 36 are also extra. So if you happen to get these correct and learn them, it will help you for college in your histology labs, but it's not required for this class. All right, we just have one more thing we're going to talk about, which is glands. So I told you that there's glands that release secretions in our epithelium that go to the outside and ones that go to the inside. And what's nice is if you took biology, as you've heard of some of this before, this kind of terminology, the exocrine means to exit or release things outside, and endocrine means to secrete it inside into the blood. So glands, because they're releasing something to a surface, are going to be part of our epithelium. So the slide I really want to talk about, though, is right here. And this is all we're going to do is make these 3 categories, so leave some room to talk about each of these types of glands. This is for our next live session. So we have eccrine glands. I don't usually use the word merocrine, but different textbooks use either one of these interchangeably. I like eccrine just because I have you guys circle the E for everywhere. So these are the ones that go all over your body. These are the ones we normally think about in terms of sweat. So these are the ones that are releasing that kind of fluid, watery fluid onto you, okay? Versus apocrine, I have you circle the A, which is for A regions. So these are found only in your armpits, your anus, and the areola, or the pigmented part of your breast. So it's found in those 3 A regions. And then sebaceous glands are also called holocrine, and those are sebum. They're oily glands. So they're secreting right onto the hair follicle to keep it moist and waterproof. And it's got other antimicrobial properties. All right, we are going to stop there though. So we'll pick up with this gland section, and then we'll move on to connective tissue next live session. And we're going to switch gears to talk about where your lab documents are. So if you're back on that stream page, and make sure you guys can see what I'm doing, and you switch to classwork, and you go to this week. I've already opened it, but you go to homeostasis lab, and then there's 3 documents here. There's the Jamboard that we did last time. So if you want to open that and reference that, you can, but we're not going to work on the Jamboard today. We're going to work on these Google Slides today. So the Google Slide one says the kind of 2 goals I have for today. Number 1, you're going to pick one of these slides, 2 through whatever, for your team. So if you already see names on it, then keep going down. Number 2, once you're in a team, you are going to choose roles. So who's going to be your techie? Who is good with graphs and charts and organizing data? Okay. Who's going to be your physiologist? Who is able to actually monitor family members and collect data? Okay. Who has the ability to do that, and the sensors, depending upon whatever it is your group decides their question is. Facilitator is your recorder, your timer, keeps your group task oriented, and records information from your team members as needed. And then your leader is interacting with me if there's any questions or problems, and then reports out. So at the end of the activity, you're going to tell us what your group finds out from your conclusions about that particular thing. So what I can do on Zoom is I can open rooms that you guys can't. It says that you guys can go in them, but you can't. But I can put you in them that are already named based on your Jamboards. So I don't know if last period said they couldn't see this, but on your Jamboard, somebody had one of the topics being something about temperature. Someone had something about homework due. Another person had mask versus no mask. Another person had one about food or diet, hydration levels, time of day, sleep. I don't think your class had sleep, but I thought that was an interesting one. And then I have just like a catch-all if I didn't put yours up here and you really want to study that particular thing, which will be room 8. So what I need you to do is tell me which of those you want me to put you in so that you can get into teams and talk about a good question. Okay, so we're only doing 1 and 2 today. You're gonna decide on a question that your team can ask about your body and homeostasis and monitor, and then decide who's gonna do what. If you end up in a really big room, like 16 of you, then I'm gonna probably divide you into 4 groups, okay, within that. You can still study the same topic, but hopefully you can come up with a slightly different question that I will look at. So Rain, which one of those topics sounds interesting to you? You can unmute.
What do you mean by topics? Where are the topics?
They're on your Jamboard. So if you click on period 6 Jamboard, the same place the homeostasis lab is, there's temperature. Somebody was curious about environmental temperatures versus their internal body temperature, amount of homework due and stress, what that does to certain body parameters. Somebody was interested in wearing a mask versus no mask on certain features of your body. Somebody wanted to know about their food and diet, hydration levels. So which one of those on that Jamboard were you interested in exploring? I'll come back to her, Rain. Katie? Heart rate? Heart rate with what? So you're going to monitor heart rate in response to what thing? lost her. Okay, back to Rain.
Sorry, it cut out for a second.
It's okay.
What did you say?
Which of these rooms do you want to be put in? Which topic?
Heart rate.
So heart rate and what though? Heart rate's your dependent variable. Which thing is going to make your heart rate change?
And your like fluids. Like
Fluids,
how much
hydration
water-
levels? Okay. Becca?
Okay, wait, I'm sorry. I'm really confused what's happening.
So on the Jamboard, we brainstorm different things that would affect your set points, your homeostasis. So you guys told me about amount of stress in your life, masks versus no mask, diets, hydration levels, all these different things that could impact something in your body, whether it's heart rate or respiration or whatever, sweat levels, doesn't matter. But which of those topics from the Jamboard are you interested in?
The full dilation one looked kind of cool.
The what one?
The like dilation of your pupils. I think that was one of them.
Oh, okay, yeah, it was. I'm gonna put that as roommate because I forgot to write that one. So, Brianna.
I don't know yet.
Okay, I'll come back to you. Briquel.
Can I do how does hunger affect your heart rate?
Okay, let's find hunger or food. I know I put that up here. There it is. Adam. Emily.
Temperature.
Okay. Elsie.
I'll do temperature as well.
Okay, Philip.
I kind of wanted to do the acidity of urine depending on hydration.
Okay. Nisa.
I want to do temperature depending on the time of day.
Okay. I'm gonna do time of day for that one. Vanessa.
I would do temperature.
Okay, April.
Temperature depending on time of day.
Madeline.
Okay, I was hoping I could do the pH of saliva based off of the foods or drinks you're eating.
Okay, I'll put you in the diet group, then you
Okay.
guys can talk about that one. Thomas.
I was also thinking pH of saliva.
Okay. Grace.
Can I do temperature depending on time of day?
Okay. Natalie.
Temperature depending on time of day.
Okay. Joseph.
Temperature.
Okay. Megan.
Can I do the hunger like affects your heart rate one?
Okay. Food. Okay. Evan.
I like to how masks affect your airflow.
Okay. Eli.
Can I do temperature and time of day, please?
Okay. Paige.
Can I do something like hours of sleep versus amount of stressors?
Okay. Let's see. Neil.
Can I do something like temperature and the time of day?
Okay. Lauren.
Can I do acidity of urine?
Okay, let's see. Let's try that one. Austin.
Also, I'll do temperature versus time of day.
Okay, Emma.
Blood oxygen, mask versus without a mask.
Adam. Adam or Brianna? We're the only two.
Can I do temperature of the time of day?
And then Adam. Put you in sleep.
Mrs. Roberts, I chose the acidity of urine one, and you put me in a group with pupil dilation.
I don't see that one as a thing. Like I don't see, there's not a separate room for it. You can still do it, but it doesn't look like there's anybody else doing acidity.
Is there one for pain tolerance?
No, I don't see that one either. The only ones I have are the amount of, wait, that has 0. Mask versus no mask, sleep, and I don't know, whatever, I think the one you were just in, room, it says roommate.
Can I do sleep, please?
Yeah. All right, I'm gonna go see.
Since our group is so big, we were just wondering like if we could split, like we kind of already know who wants to be in what.
Yep. So all you have to do is go and choose a different Google slide.
Oh, okay.
And then put your names on that one and then tell me which question you're gonna do about that topic.
Okay. So yeah, me, Nushi, Grace, Eli, we're doing the how is temperature affected by the time of day.
Okay, yeah, so just decide on your roles and write that question. That's all we're trying to get done today.
Okay.
Okay, thank you.
Okay, I saw you guys had a question.
Yeah, so we like, we all like, two of us, like, between Madeline and the sitting, then me and Megan have like what we chose, so we all supposed to pick one together. Are we doing it like separately or because they're different like responses?
I can only, whoa, I can only hear, oh, my sound's bad too.
Can you hear me?
Yes.
Okay, I can explain. We have the same
Okay.
here. So like we have two different like experiments going on. So like Brickell and Megan have one, and then me and Tommy have another one. So we were a little confused if like we're supposed to like work in groups of 2 or if we're like supposed to somehow.
You can work in groups of 2, just pick a different slide. So what you're doing today is picking your slide with your partner then and write both your names on that and then tell me what question you're gonna do. So what question are you guys, like how are you phrasing your question?
So we're doing how consumption of different food and drinks impacts pH of saliva, and they're doing how hunger impacts heart rate.
Okay. Yeah. So just write that out and then kind of tell me who's going to do what roles, since you're going to have to take on more than